And if Dr. Polson can be available and go ahead and come on screen, he is an associate professor in the departments of Computer and Information Sciences, Plant and Soil Sciences, and Biological Sciences at the University of Delaware. His research interests lie at the intersection of genomics and microbial ecology, examining the ways in which microorganisms and their viruses affect and are affected by their environments. In light of COVID and other viruses, this could be a very relevant topic for our world. Dr. Polson, you are on the stage.
All right, great. And can everyone hear me?
I can hear you fine, and your presentation is showing fine, so it's all yours.
Excellent. All right, well, thank you to the organizers for the invitation to talk today. I put together a talk that dives a little bit into my research and actually kind of looks at a bioinformatics education thread with undergraduates that has kind of shaped my research program over the years, and we'll get into that in a little more detail. But as Grant mentioned, I am at the University of Delaware, and I wear a few hats there. In addition to my faculty role, I'm also director of Bioinformatics core facility and the Bioinformatics and Data Science core aspect of the Delaware INBRE program. So I work with investigators pretty much across the swath of biomedical data science and bioinformatics in trying to figure out how to interpret data that they have, how to actually move forward their research using different tools, and how to educate people in bioinformatics. So a lot of what we saw in our last talk was great, really kind of hit the nail on the head with a lot of the topics that I'm usually talking about in my presentations. So as I said, I'm going to talk a little bit about my science today, but I did want to briefly mention kind of our bioinformatics data science infrastructure at the University of Delaware. I think the University of Delaware was really forward-looking about 15 to 20 years ago and making a really big investment in bringing in a strong bioinformatics aspect to the campus. And one of the unique aspects our bioinformatics programs have is that our campus, we are not associated with a medical school, which actually means that we have a lot of life science research that is going on that is not only in the biomedical sphere, but goes beyond that to agriculture, biopharma, bioengineering, chemical engineering. So there are just tons of related disciplines that end up feeding into our bioinformatics center that I am part of. So we have a really vibrant kind of population of people that are interested in omics, data science broadly, and our center, which has been around for about 15 years now, is both holding those core facilities that I mentioned that I host, and also a really strong educational program where we've developed a number of different graduate degrees in bioinformatics, data sciences that we have dozens, hundreds at this point of students have passed through our various graduate certificates, master's programs, and of course doctoral level programs. And we're really engaged in education throughout that entire realm. But one thing I did want to include in this talk is we are really excited that our already what we feel like are really good bioinformatics education programs have just gotten a huge boost from an NIH investment, which I am lead PI on this new T32 graduate training grant in computational biology, bioinformatics, and biomedical data sciences that we're going to be hosting at the University of Delaware, and actually just gave the launch orientation for that a couple of days ago. We're really excited about the opportunity to bring this in. It comes with a number of new slots for students that we can fund and bring into our program, but also is going to let us really work in those kind of interesting experiences with working with industry, working with government, and working with people in the medical industry as well. So I just wanted to put in a plug for this. We are recruiting for our next cohort starting next year. Anybody who's interested in our program, please feel free to reach out to us at the bioinformatics@udel.edu email address. So without further ado, on to the research talk that I wanted to give for today. So my entire career has been, based around looking at microbes and what they do. And I think, we've seen in the past, decade or so that the role of microbes, the microbiome associated with humans and other organisms, they've really started to take a much more central role than they ever had in the past. So if we look at this kind of extent of genetic diversity on Earth, and we look at this tree here, you can see that this little red circle at the bottom, pretty much every organism that you can see with your eye is within that red circle. All the rest of this diversity, it's microorganisms, from bacteria to archaea to microbial eukaryotes. So it really is it's a microbial world. I don't think this is any secret. But even this tree does not capture that full extent. There's obviously one missing piece. What is it? Well, it's viruses, right? So viruses obviously have been very much in our consciousness the last couple of years with the COVID epidemic. But I tend to research more on a different group of viruses, which are the microbial viruses, which would be any virus that infects one of those microorganisms that I mentioned. And these are, by far the most diverse group of biological entities on the planet. If there is a gene on this planet, probably a microbial virus is carrying around some version of that gene. And they're probably one of the most poorly or least poorly understood groups of biological entities. I always hasten to call them organisms on the planet. And that's a lot of what my research has been based around, is how to figure out ways to understand viruses and how they impact their environment. So I have a joint research group here at the University of Delaware. I've actually teamed up with two other faculty members, Eric Womack and Jeff Furman, and we've developed a joint group that looks at different aspects of these virus, microbe, environment interactions. And that environment, really, it could be anything. I have looked at, this is just a small selection of things I have looked at, but it could be anything from, you see seagrass here, corals, seawater, but also humans, right? Humans are an environment, and a major environment for microbes. So looking at humans and biomedical models, agricultural models, you can see soybean here. These are the types of projects we do, but what unifies them is this interest in the microorganisms, how those microorganisms interact with these either environments or eukaryotic organisms, and then ultimately how the viruses that infect those microbes play into that. So it's kind of a tripartite system we're looking at. And to study this, obviously DNA, those types of omics techniques are the bread and butter of looking at these types of systems. So those are really kind of the unifying themes that go through my research. So, just as a basic background, viruses are really difficult to study. If you can go out and isolate a virus and sequence its genome, you're golden. You can do a lot of research with it, but it is very, very hard to do that. Even when we know what the host is, say human, it is often very difficult to go out and find specific viruses that are infecting that host, much less if you're looking at environmental samples where there could be thousands of potential hosts and thousands or tens of thousands of different types of viruses, and figuring out which virus goes with which host, and what can you culture the host? Can you culture the virus? So there's a lot of complexity in trying to study viruses. So DNA-based methods have really become important for looking at viruses. However, a lot of the tools that allow us to use omics-based techniques in other organisms, like marker genes that are universal, say the 16S rRNA gene that gets used in bacteria, don't exist in viruses. There is no one gene that every virus has. So it makes it quite difficult, quite complex to look at. So using shotgun metagenomics, which would be going out into an environment, taking a sample that has a broad mixture of organisms in it. Typically what we'll do is we will filter out the cellular organisms into a separate fraction so that what's left is just things of a size that should be viruses. And then we sequence that entire sample, right? So we get a huge amount of DNA sequencing data back, and it's about many, many, many different types of viruses that might be living in an individual sample. So then we use various techniques to go through and look at that. And some of those techniques can be quite computationally expensive, but as computing and other technologies have advanced, that's really not a limiting factor in doing these types of techniques at this point. At this point, really our limiting factor is our ability to make biological interpretations out of those genomic data that we get. I'd say in viruses, it's really hard because, as I mentioned, you can't culture many of them. So for us to actually have phenotypes in the viruses that we could potentially correlate with things we're seeing genotypically in that metagenomic data, it becomes a real challenge for us. So, other issues that we run into are things like, if we look at any given environment and we metagenomically sequence it, and then we cluster all the putative proteins that are there, and we look at them from the most common to the least common. Among the most common proteins, one study we have here, we found that 62 of the 100 most abundant putative proteins were uncharacterized. And of the ones, which is saying they were not in a genome at that point, and of the ones that were actually in a genome, all the ones that have arrows pointing to them here were, they were annotated as putative uncharacterized protein, which might as well be uncharacterized or unknown for all intents and purposes. So this is a major wall we also run into, is that, there's a lot of genomic data out there, but we don't even know what gene it corresponds to, so much less what type of activity it may have. So it's been an ongoing challenge, and probably one of the things that keeps me interested in this work is that it's challenging to go through these data and figure out ways to make meaningful biological conclusions based on them. So I mentioned, undergraduate researchers have played a huge role in shaping this research program. And this is actually a slide from about 11 years ago. This is a program that we've had going on for quite a while. But what we started to do with my collaborators was to each summer recruit a cohort of undergraduate researchers into the lab. And we're lucky to have excellent summer undergraduate research programs at the University of Delaware that have helped us to do this and to bring these students in. And we've really put them to work on doing metagenomic analysis, on doing some of the lab-based steps downstream of it, so that we can start to understand a little bit more about this diversity that is out there. So one of the main approaches we've used with them is we'll take these protein clusters that we see in metagenomes, we'll have them explore them, we'll have them adopt a particular cluster or a particular type of cluster. I have ribonucleotide reductase clusters kind of pointed out in this graph here. So one undergraduate researcher may take these, and they will actually go through the metagenomic data, pull out the sequence reads for those that contain those genes, do assemblies, do phylogenetic analyses, ultimately build some primers so that they could look at this and try to actually identify it in environmental samples. And then, take some environmental samples that we have in our library and see if they can actually re-identify these genes in those natural samples. And through doing this, it really increased our bandwidth to be able to look at all that metagenomic data that we were holding on to and really start to understand some of these genes that we see very commonly in the various metagenome sequences. And we've definitely looked at both unknown and known functional genes through these types of methods. And we have interesting stories on both. Today, I'm going to follow through with one story that is from a known gene, which would be DNA polymerase 1. So if you're not familiar, it is the proofreading polymerase in many bacteria, but in viruses that carry it, it is the main polymerase. It is the one they use for replicating their genome. And it is a fairly common gene for a double-stranded DNA virus to carry. We see it in about 25% of known DNA viruses, double-stranded DNA viruses. So that's actually a pretty good percentage for us. So looking at this gene gives us the ability to look across a lot of different viruses. So one group that has looked at this, and there are a couple of grad students, Jess Chopik and Dan Nasco, also Eric Sikowski, who's not on here, and in particular, one undergrad, Helen Schmidt, who really adopted this DNA polymerase gene over the course of several years and made some really interesting discoveries with it that I wanted to walk you through. So first is a little bit of background to remind you, viruses and microbial viruses in particular, tend to come in two lifestyles or flavors. There are the viruses that will enter a cell and immediately they're going to go into the replicative cycle, make a lot of copies of themselves, and immediately lyse the cell. So those are the ones that are virulent or live the lytic lifestyle by default. The other group are the temperate phage or temperate viruses. Those are the ones that enter the cell, incorporate themselves into the host genome, will lie dormant for some period of time, and then eventually some trigger in the cell or the cell's environment will cause it to essentially activate itself and then do a replication. So understanding which viruses are lytic and lysogenic is a pretty important thing when you're trying to understand viral biology and how viruses impact their hosts. So it seems like this would be a very easy thing to identify within a sequence, but it's not always. And actually, many phage that are out there, we have complete genome sequences, but we still don't know for sure which one of these lifestyles those phage tend to follow. So Helen Schmidt, as I mentioned, she was an undergraduate biochemistry major. And she came into one of our summer undergraduate internship opportunities and adopted this DNA polymerase I cluster and started looking at it in much more detail. And through her work and her particular interest in biochemistry, she noticed something within this gene that has led to 12 years of subsequent research for us. And that was the fact that within this gene, when you look at the protein sequence for it, there is an individual amino acid residue within it that seems to be predictive for the phage lifestyle. If we look at this residue at position 762 relative to the E. coli version of the gene, the default that you tend to see in bacteria is a phenylalanine at that position. However, if tyrosine is at that position, there is biochemical data out there that indicates that makes the polymerase about 1,000 times more processive, faster, essentially, but also makes it more prone to errors while it's replicating. There's also a mutation that's been biochemically characterized to leucine at that location. And when that amino acid is in there, we get about 3 times more accurate, but about 1,000 times slower than the phenylalanine. So it's quite a spread in the speed and the accuracy that occurs based on this one amino acid change in polymerases. And what Helen noticed was that this individual amino acid residue, when she overlaid its identity onto a tree, very neatly clades with our groups with the clades that were on the tree. We can cleanly see there are 2 groups here that are leucine, but there's a single group each for phenylalanine and tyrosine. So to us, that was an indication, wow, there is a real biological phenomenon going on with this amino acid residue. And we really saw that when she started to look at the lifestyle of the phage that we did know their lifestyle. And that was that the tyrosine version was always lytic. We had no exceptions at the time of a lysogenic phage that had tyrosine. Phenylalanine was almost always lytic, a few exceptions there. And leucine was almost always lysogenic. So just this individual residue gave us that information that otherwise is very, very difficult for us to identify out of these types of sequences. So if we just do the thought experiment of why this might be important to viral biology, let's think about, lytic viruses are the ones that come into their host, they replicate fast, they produce lots of copies of themselves, and then they're out of the cell. So for those guys, if they have errors in a few copies, maybe that's okay. Maybe occasionally you're going to get a mutant, and maybe that mutant's actually a good thing, right? Maybe that's new diversity that's going to allow them to succeed. But if that mutant actually is bad, then, it's okay. You've got lots of copies that are faithful to the original. And occasionally you're going to get a really crazy mutation, which, may or may not be a good thing, but you have to see how that works out with natural selection. But the really interesting thing with this trait is you're modifying your polymerase, and your polymerase is ultimately responsible for the mutations that could lead to natural selection, lead to evolution. So really by modifying their polymerase in this way, or naturally selecting for the polymerase in this way, they're evolving to increase the rate of evolution. which, that's a phenomenal thought, right? They're really giving themselves the ability to be more diverse. If we look at the other side of the coin, temperate phage tend to replicate slower. They're sitting within their cell for a long time, so they really need to be more in sync with the cell. And then when they actually do start to replicate themselves, they're doing it at a critical time when there's probably a stress on the cell, and they really need to get accurate copies and get out so that they can survive and reinfect a new cell. So in those phage, selecting for a more accurate mutation, this leucine mutation, even though it's a bit slower, may have significant evolutionary potential for keeping it in sync with its host. So for the rest of my talk, or actually one more slide on this. So the first students I talked about, they were doing that whole process very manually. We've actually since done a lot of work on streamlining it, making it easier, building bioinformatics tools. I've had a number of grad students who have come through, and we've just made an entire suite of bioinformatics tools that make this process of investigating these viral genes in this way very straightforward and easy to do. So for the rest of the talk, if we go back to this DNA polymerase A, we've looked at plenty of other clusters, but this one has really been one that has really captured our interest. And I mentioned this 1,000 times faster, but not quite as accurate, or 3 times more accurate and 1,000 times slower. Those are really, fairly widespread in biochemical literature. But when we really traced it back, they all went back to single studies where they tried some in vitro mutations. So really, we didn't know for sure that all polymerases followed this rule, or that it's something we would actually see in the environment. So a current study we have ongoing now is actually taking the bioinformatics back into the lab. And I have a student, Rachel Keown, who's been working with some collaborators at Viruse Biosciences. And what we've done is actually taken our metagenomic sequences for DNA polymerase A and have picked out different variants of them across the tree, represent these different types of amino acid residues that come from different sources. And we've had them synthesized, had the DNA synthesized, and then clone those into expression vectors so that we can actually produce the protein and do biochemical characterizations of it. So this was something a little bit out of our wheelhouse, and we were really hoping it was going to work. And fortunately, working with these experts at Viruse, it's working. We have actually taken, we've had about 50 of these synthesized at this point, and we've had about a 60% success rate of being able to isolate a protein out of it, and that protein actually having functionality. So it's really cool that we can actually take a sequence from a metagenome and make a protein out of it. But what we're ultimately interested in is we want to find viral phenotypes, and the biochemistry of these enzymes ultimately determine how fast that virus can replicate, how fast it can copy itself and get out of its host cell. So understanding the different phenotypes biochemically for this enzyme is something really important that we can now layer onto other data that we have. So, I won't go into explaining this figure because I'm getting a little short on time, but the bottom line is, if we look through these different ones that we have synthesized, we have found quite a few of them that we can show have primer extension activity at various speeds, various rates. We also have several of them that have a strand displacement activity, which is an interesting activity on its own that our partners in industry are very interested in, but we also are interested in is that it's another phenotype that organism has, or that virus has, that it's carrying around that we can potentially look at. So Rachel is moving forward with this work. She's gone a little bit beyond only looking at this biochemical data that she's been producing, but she's also looked more at what else goes with the polymerase. It's not living in a vacuum. It's ultimately working with other genes to replicate the genome. And we really see that there are some really constant associations between our different tyrosine polae, phenylalanine polae, and leucine polae, with different types of helicase genes that are specifically tuned to work with individual ones. So this is another piece of evidence that this processivity accuracy hypothesis that we've been testing may well be true, in that GP4 helicases, for instance, are known to be among the fastest helicases out there. And if you're unwinding the DNA for a very fast polymerase, you would think you're going to need a very fast helicase. So that we're seeing these associations is very promising, and we're now starting to move beyond the pol A and also look at the biochemistry of these accessory genes like helicases, like ribonucleotide reductases that we see in association with these, and look at that broader genome neighborhood and figure out how that module of genome actually impacts the biology of the phage. So where are we going with this work? Ultimately, Rachel's still working on purifying and biochemically characterizing those various enzymes. She's worked with another group of undergraduates this summer in doing optimal pH and temperature assays, which can actually tell us a little bit about the biology of the cells that the phage are infecting based on where their optimal amplification or where their optimal operational temperatures and pHs may fall. As we're expanding this database of biochemical information about different phage from different phylogenetic parts of the tree, we're correlating that data with other information that we either have based on where they were isolated or abundance and infectivity information that we're producing in other areas of our lab through various collaborations. And our ultimate goal here is we're going to take this massive data, both on biochemistry, observable phenotypes when we can get them, as well as the genotypic features that seem to correlate with these. And we're going to integrate them together and begin to see if we can make models that will allow us to predict phenotype more accurately based on the genotypic features that we see within these replication module proteins, and how well it can interpret or predict these types of infectivity characteristics or environmental characteristics that may apply. And we've done this on some fairly simple basis so far, just looking at combinations of ribonucleotide reductases and DNA polymerases that we see occurring together. And we've actually had some really interesting results come out of that, where we can see correlations between versions of ribonucleotide reductase that require oxygen versus don't require oxygen, and the available oxygen or metal cofactors that are available in their system. So we have some great preliminary data here, and we're really hoping to build a more substantial model moving forward with our work. So I think I am wrapped up now. Thanks to everyone in my lab, my collaborators, and of course our funding sources.
Thank you, Dr. Polson. That is, again, fascinating stuff. If you don't mind, you can quit sharing your screen so we can see you. How are these in silico tools like AlphaFold for research you're working on? How useful are they?
They're actually quite useful. I actually have a grad student, Zach Schreiber, and a collaborator, Madi Belkade at the University of Hawaii, and they are looking at particularly at protein structure that we can predict with these new in silico AlphaFold 2 and so forth, and how well that can be used to classify these proteins in metagenomes. So that's actually a different direction we are going, but it is something we are investigating in this area.
Very fascinating. How important is mentorship and student participation in this research? You've mentioned a whole bunch of names that aren't yours during this.
Yeah, absolutely. So, student participation is, our research could not happen without it, both undergrads and grads. And we really try to set up a kind of a group support mechanism. We don't just take an individual student through, we bring through a cohort of students, which brings a support mechanism with it in itself. And then we pair those undergraduates with graduate students. And then we have mentor PI meetings with them on a regular basis. So through that kind of multi-level mentoring, it both teaches graduate students to be mentors, but it also sets up multiple levels of support for those students as they move forward to get at these types of new skills that they're trying to learn and apply.
Thank you so much. I've wrapped up our time with you, and we really appreciate that. I think we could ask questions for another half hour if we could, but we need to move on. So thank you again for your participation here this afternoon. We really appreciate it. Thank you.
Thank you.